need to be achieved under the constant conditions, including the pH, the temperature, the ion concentration. So to ensure that the normal physiological response is occur, the animal needed to maintain the constant osmolarity and ion concentration inside their body. Okay, so make it a very easy understanding the case. So this is a iguana. So they is a big lizard live near the sea, and they probably drink a lot of the seawater because there are no fresh water in their living environment. So because of this, so they basically accumulated too much sodium chloride inside their body. So in their, on the top of their head, so you can see they excrete the sodium chloride on the top of their head. And this is because they have a special organ called salt gland. So this is the diagram of the salt gland. So the epithelial cell, they expressed a special transporter which can transport the sodium and the chloride to the lumen of the secretory tube. Then the excess salt can be excrete inside outside of the body. So here, that is the way they evolved to adapt the seawater environment. So regarding to the ionic and the osmotic challenge, so the terrestrial animal and the aquatic animal, they are face exactly different challenges. So in marine environment, animal tends to gain salt and lose water because of the difference of the osmolarity. In contrast, in the freshwater environment, the animal tend to lose salt and by perfusion. Not perfusion, sorry, diffusion, and again water because of the difference of the osmolarity. And the terrestrial environment, the animal tend to lose water because water is difficult to get compared to the aquatic environment. So many animals move between environment and must be able to alter their homeostatic mechanisms. For example, some aquatic animals, they can live in the seawater and the freshwater. And these are two different habitats. They need to change their strategy to maintain their osmolarity and the ion concentration inside the body. So in the terrestrial animal like mammalian, so here is the human kidney as an example. So the human kidney, the unit of the kidney is the nephron. The nephron consists several tube systems, including the proximal tubule, loop of Henle, and the distal tube, and the collecting duct. So the different parts of the tubule, they can excrete or reabsorb the ion. And by regulating the excretion and absorbing the ion, the mammalian like humans, they can actually maintain the constant osmolarity and ion concentration inside the body. So in terms of osmoregulation, there are two main strategies among the animal kingdoms. One is the osmoconformer. That means the internal and the external osmolarity is similar. For example, the most invertebrate. So they basically do not regulate their osmolarity. And this strategy is energy saving. But at the same time, because of their osmolarity inside the body very different, differ from the environment they face. So they have to evolve a different strategy to overcome the changes of the osmolarity because they need to maintain the water content inside the body. And the other kind of animal called osmoregulator, that means they can maintain their osmolarity constantly by active transport. So in the freshwater region, they need to uptake iron. And in contrast, in the seawater environment, they will excrete the salt actively. So here I show you two examples. One is the freshwater fish, and the other is seawater fish. Seawater fish and the freshwater fish. So as I mentioned, seawater fish, they tend to lose water and gain too much salt. So they need to overcome the loss of water. They need to drink a huge amount of seawater. But at the same time, because the seawater contains too much salt, so they need to excrete the iron actively. So they will produce the higher concentrated urine, and they will actively excrete the iron from the gill. So in contrast, the freshwater fish, they have a different challenge because they tend to gain water and they tend to lose the iron. So they need to drink very few water, but they need to actively uptake the iron from the gill, and they need to produce huge amount of volume of the urine to decrease the water content inside the body. So the fish gill is the most important organ to maintain the osmolarity and the iron concentration inside the body compared to the terrestrial animal. So the mammalian like us, so we use the kidney to perform this function. But in fish, the most important organ is actually gill. So the water flow goes through to the mouth and then perfuse to the gill. Then in the gill, the fish actually breathe, uptake the oxygen and dissolve the CO2. And at the same time, they perform the iron regulation inside this part. So if you look the structure and the cell types of the gill, you can see the filament and the lamina. So the lamina is the most important part for fish to perform the respiration. And in the filament, you can see so many cell types. So these wavy convex, shallow basin, and the deep hole. So this is the cell type for the iron and the osmoregulation, called a chloride cell or mitochondria-rich cell, or you can say it is an ionocyte. So the structure of the ionocyte is like here. So this is a pavement cell, basically for respiration, and the chloride cell, they have many mitochondria. So you can also call this is a mitochondria-rich cell, and they are big. So they have the apical and the basolateral side. So this is a SEM. So this is a pavement cell, and this is a chloride cell. So the function of this cell is basically for the acid base or ion regulation. So in this experiment, you can see the size of the apical opening. So under the pH 4, the opening is significantly increased compared to the pH 7 and the pH 10. That means this type of the cell, they are responsible for the acid excretion. That means they excrete the proton. So by using the immunohistochemistry, we can see at least two types of the ionocyte or mitochondria-rich cell in the body, in the gill. So one is the red, labeled by red, called sodium potassium ATPase rich cell. The other is a proton ATPase rich cell. So they are different types of the ionocyte. And over here, you can see the proton pump rich cell. Okay, it is very important for acid-base balance. So to date, fish already became a model for study the ion and osmoregulation because the gill is very easy to assess. So different strategy can be used. For example, the transcriptome screening or DNA library, and the mutant fish is easy to get, and the double triple colocalization of mRNA or protein is doable. And the most importantly, the gene manipulation method is also easy to apply to this species. So to date, at least 4 types of the ionocyte have been identified. They are proton pump rich cell, sodium potassium ATPase rich cell, and the sodium chloride cotransporter rich cell, and the other is a potassium sensitive cell. So they are responsible for the different ion balance. So for example, the HR cell, they express the proton pump in the apical membrane. So this cell is basically functions as acid-base regulation because they can pump out the proton actively. And in contrast, the sodium pump-rich cell, they are responsible for the calcium uptake. So they express the epithelial calcium channel in the apical membrane. So the calcium enter to the cell passively because inside the cell is the cell calcium concentration is very low. And after the calcium enter to the cell, they immediately bind by the calcium binding protein, then transport the calcium into the basolateral membrane. Then the plasma membrane calcium ATPase or sodium calcium exchanger located in the basolateral membrane, they transport the calcium inside the body. So the sodium chloride cotransporter rich cell, they are responsible for the sodium and chloride uptake. For the potassium sensitive cell, the function of this cell is to exclude secrete the potassium. And interestingly, so these 4 types of the cell can be found in the mammalian kidney cell, called collecting duct cell, DTT cell, or intercollected cell. So they have a similar function. So if you want to study the kidney function, so for the ion regulation or osmoregulation, the fish gill probably can be the alternative model. Compared to the mammalian kidney, the fish gill is relatively easy to manipulate. Okay, I will skip this. Okay, so by studying the osmoregulation, the morphology and the function of the ionocyte is very important. For example, there is a type of fish, they can live in the seawater and the freshwater environment. So this is a salmon. So we can see if you stain the sodium potassium ATPase, as I mentioned before, so this type of the cell is responsible for calcium uptake in the freshwater fish. So, but as I said, the freshwater and the environment, freshwater and the seawater, the environment, they are very, very different in terms of the ion concentration and the salinity. So in the seawater, if you stain the sodium, potassium, ATPase, you can see the sodium pump-rich cell, the morphology is very different. So they have a large cell, and compared to the fish live in the fresh water. So here is the diagram for the chloride cell function if they want to excrete their sodium and the chloride. So in the apical membrane, they express the ion channel called a CFTR. So it is a chloride channel. And the basolateral membrane, they express the sodium, potassium, chloride cotransporter. So they will use the sodium gradient to transport the potassium and the chloride inside the chloride cell. And then the chloride diffuse by the gradient to outside the body. And for the sodium, so they use the cell-cell junction passively leak out the body. So apart from them, so this type of cell, because they have many transporters, and they need to consume ATP. So another feature, a character of this cell is they have many mitochondria. And of course, they have many sodium, potassium, ATPase. So if you look the detail of this type of cell, you can see so the fish transferred from the fresh water to seawater for 6 days, and you can see their morphology of the ionocyte is very different, as I said. So if you stain the NKCC, sodium, potassium, chloride cotransporter, and the sodium, potassium, ATPase, you can see a very large cell in the seawater, but smaller in the freshwater cell. So our lab recently published one paper, and we found that there is a kind of a fish famous in Japan called medaka. In Chinese, is kind of 大肚鱼. So the Japanese medaka can live in seawater and the freshwater. And we found that if we pre-exposure to the seawater, put the medaka to the seawater for 1 week later, then that will dramatically increase the survival rate during the seawater acclimation. So here you can see the Japanese medaka exposure, pre-exposure to the seawater. Then we transfer them to the seawater again. So if we don't treat the pre-exposure like training, so they 100%, so they cannot survive in the seawater transfer, directly transfer to the seawater. But if we trained the fish as brackish water, then this survival rate will dramatically increase to more than half fish can survive in the seawater transfer directly. So we analyzed the gene expression pattern of this seawater retention gene. We found that several genes is called as ATP1A1A.8 or ATP1B1B. So this gene, they are consistently or sustain the expression in the fresh water. So we start to sample the fish gill in the fresh water, and then transfer to the seawater, and then transfer back to the fresh water. We found several genes that like these two, they can sustain their higher expression, even the fish transfer back to the seawater. And we think that probably this retention can increase the survival rate. So when the fish go to transfer to the seawater again. So we performed the double in situ hybridization, and we demonstrated that those two genes are actually located in the ionocyte. So we use the sodium potassium ATPase as a marker. Okay, so when we try to assess the function of the ionocyte, we use the fish embryo, because the ionocyte in the embryo, fish embryo, they are located in the yolk sac skin of the yolk sac, because during this period the gill is not developed very well. So the function of the respiration and the ion regulation is performed by the skin. So we apply the electrophysiology for investigating the ionocyte function. So the principle is like this. We use the very tiny glass pipette as a probe. Then we move very close to the embryo or out or away from this embryo. Then we calculate the ion concentration difference between this microenvironment. So by this, so this is a mitochondria-rich cell. So this is a glass pipette. So when we move near or far away from this cell, we can calculate the concentration difference of the specific ion. Then we can measure the ion influx or ion efflux. Like this. So by this method, we are successfully to measure the ammonia excretion and acid excretion and the sodium uptake function. Okay, so the final part I want to explain is how does the ionocyte function be regulated? So the answer is hormone. So the most famous hormone is vasopressin. So vasopressin is a peptide hormone, very similar to their good friend called oxytocin. Okay. So the vasopressin receptor is a kind of the G protein coupling receptor. So once they bind to the vasopressin, they will activate the signal transduction inside the cell, then activate the protein kinase A, then to promote the membrane translocation from the ER to the membrane. So this protein is called aquaporin for the water uptake. And the other hormone is like a cortisol, okay? So they can pass through the cell membrane directly and regulate some ion transporter genes, expression of the ion transporter genes. Then this ion transporter can be transported to the cell membrane to increase the ion concentration function. So to date, our lab, not our lab, so my lab and my collaborator, we have already identified several hormones that can regulate the ion regulation. So for example, this is isotoxin in fish, but now the name is called oxytocin in fish. So the oxytocin can promote or increase the cell proliferation of the ionocyte. And steniocalcium, so the student will present the steniocalcium today. The function of the steniocalcium is basically inhibit the calcium uptake and the cell proliferation of the ionocyte. So the parathyroid hormone also involved in the mRNA expression of the transporters and the cell proliferation. And the other hormone like calcitonin and vitamin D, so they are all involved in the calcium uptake. So by regulating the proliferation of the stem cell or differentiation of the ionocyte, they can control the cell number of the ionocyte. Or they can they can regulate the gene expression of the transporter. Then they also can regulate the decrease or increase the activity or expression of the transporter, then achieve the function of the ion regulation. So I will make an example for describe the hormone regulation of the ionocyte function. So first, we use the oxytocin as a candidate. So we treat the fish in fresh water or the ionized water. So that means the ion concentration is very low. Then we found that the oxytocin content is significantly increased. Then when we use the morpholino to knock down the oxytocin expression, protein expression, then we found that inside the body, so this is a zebrafish embryo. So inside the body, the chloride, calcium, and the sodium content is significantly decreased. That means that these types of the manipulation makes the fish cannot maintain their normal function of ion uptake, including chloride, calcium, and sodium. So that is very important for their development process. So when we try to measure the number of the ionocyte, we found that the 3 types of the ionocyte. So this is ATP6V1A. So that is a proton pump-rich cell, sodium pump-rich cell, and NCC cell. They are all significantly decreased. And when we check the numbers of the ionocyte, they are all decreased by the knockdown of the oxytocin gene, but increase after we inject the cRNA, messenger RNA, into the embryo. So indicate that the oxytocin actually, they can regulate the number of the ionocyte. So we also check the oxytocin receptor expression. So this is adult glial section. So we found the oxytocin receptor and the oxytocin receptor-like. They are all expressed in the 2 types of the ionocyte called the proton pump and the sodium pump-rich cell. So that means this ionocyte, they can receive the signal from the oxytocin and then increase their function. So finally, we want to ask if the oxytocin regulate the ionocyte differentiation. So we checked the differentiation of the ionocyte. We found that they are coming from the epithelium stem cell. Then this cell will become an ionocyte progenitor. Then under the terminal differentiation, the ionocyte can become the sodium pump-rich cell and the proton pump-rich cell. So during this process, one gene, so FOXI3A, is very important. It's a kind of a master regulator for promote the terminal differentiation of the these 2 types of ionocyte. So we inject the morpholino to knock down the oxytocin, and we inject the cRNA to overexpress the oxytocin gene. And we found after we inject the morpholino to knock down the oxytocin genes, the density of the FOXI3A positive cell, that means the ionocyte progenitor cell decrease, but increase after RNA injection. So regarding to the epithelial stem cell, so we found the similar result. So the stem cell is decreased after knocking down the oxytocin, but increased after the cRNA injection. So that means the oxytocin is important for the cell proliferation and the terminal differentiation of the stem cell and ionocyte progenitor cell respectively. So one case is that, so if we put the fish, so zebrafish is actually tropical fish, so that means they like to live under the 28 Celsius degree, and they do not like cold, like 12. So if we put but they actually can live under such a low temperature. If we put the fish into the cold environment, so here you can see the ECAC cell, okay, the sodium pump rich cell, basically expressed in the lamina, sorry, not lamina, filament in the gill, but proliferate even in the lamina cell. So that means they increase the expression and the cell localization of sodium pump rich cell. So that is because under the cold environment, the ion uptake function is strongly inhibited by the cold, because the active transport or active uptake of the ion need to performed by the transporter, which consume ATP, and this reaction is temperature dependent. Okay, so we then try to measure the mRNA expression in the brain, and we found a set of genes which is related to the oxytocin differentiation. They are all increased after the cold treatment. So when we check the oxytocin cell number, we found that after 7 days treatment, the cells number increased. And when we use the CFOS as a marker for check the neural activity, we found that after 3 hours transfer to the cold environment, so the oxytocin neuron is activated, suggesting that from the 3 hours transfer, the oxytocin still start to secrete into the circulation system. So we also check the plasma level in the fish. So we found after 3 hours transfer, the oxytocin release is actually increased. Okay, so as I said, the oxytocin receptor is expressed in the gill, and specifically saying it is expressed in the ionocyte. So we then use again electrophysiology to monitor the cell function of ionocyte. So we try to measure the proton excretion function. So we found that after cold treatment, so the excretion of proton is significantly decreased, but rescued or increased after we apply the oxytocin agonist. So that means oxytocin itself is not only to regulate the cell number of the ionocyte, but also they regulate the function of the ionocyte. So when we use the tetanotoxin system to silence this cell, we found that the tetanotoxin silence the oxytocin neuron, actually abolish the increased expression of the transporter inside the ionocyte. So indicating that involvement of the oxytocin on ion regulation. So the conclusion is that, so when we put the fish from the normal temperature to the low temperature, then the cold, the temperature sensing structure send the signal to the hypothalamus, then increase the some transcription factors expression. Then this set of the transcription factor will activate or facilitate the oxytocin neuron differentiation. Then because the oxytocin neuron, they can secrete more oxytocin to the circulation system. Then in the glial site, the ionocyte, they express the oxytocin receptor. So after they bind to the oxytocin, the signaling pathway promotes the ionocyte differentiation. Then the ionocyte will increase, and also for the transporter, the expression in the ionocyte, then the ionocyte function will be increased. And this process can be called acclimation. Means that increase the iron uptake and the acid balance regulation. Okay, I will stop here. And if you have any question, I'm very happy to discuss with all of you. Thank you.
You guys?
Okay, please.
So I wonder if the iron uptake or acid-base regulation do anything to help the fish survival with the low temperature, because due to the low temperature is
Sorry, it's
okay. Sorry. I have a question back to slide 28, 29. So thanks to the vasopressin, we have the presence of aquaporin. So I wonder if this process be reversible. So if we don't have the vasopressin anymore, is that aquaporin still maintained in the cell membrane?
Oh, thank you for your question. So the translocation of the aquaporin is actually very quick. So like 10 to 15 minutes. So if the cell sends to the vasopressin, then they will promote this reaction very soon. But also the recycling of the vasopressin is also very soon. So once no vasopressin signaling, so the endocytosis will occur, then recycle the aquaporin protein.
So when we don't have the vasopressin anymore, the aquaporin will be...
Yeah. Then you will try to pit.
May I also ask a question from Kyoto University?
Thank you. Please.
Yes, okay, I hope you can hear me.
Yes.
So I was wondering, you were talking about that in fish, the ionocytes, for example, they increase in size. And I was wondering if you know if something similar happens in kidney cells. So for example, if there is more salt in the bloodstream, if the cells in the kidney cells also increase in size?
Okay, thank you for your question. So actually, so the short answer is I don't know, because I do not study the mammalian kidney so much. But as my knowledge is, so it is quite different to see this change, because compared to mammalian, so the fish cell, especially in the gill, so the flexibility is very different. So fish can easily to change their cell type and the morphology. But in mammalian, especially in the kidney, I think probably it is difficult. So they in mammalian system, as I know, so they have already prepared several types of the cell. So the different cell type, so they are responsible for the different functions, not like a transformation of the cell's morphology.
Okay, thank you.
Thank you.
One funny question from Kyoto.
Thank you.
You talked about both Medica and zebrafish. And zebrafish are primarily freshwater.
Yes.
But can they adjust to slight variations in salt concentration
Yes.
the same way Medica do? It's just harder to see it. It's more subtle.
Sorry, so what is your question?
Can they adjust to slight modifications in salt the same way that Medica do?
Okay. Thank you for your question. So actually, we performed some experiment that, as you said, so zebrafish actually they can survive under the lower salinity environment, like 10 ppt. So in the seawater, so the salinity is 35 ppt. And we actually treated the fish inside the 10 ppt, and they survived. They probably well, because I have tried for 1 week, so 1 or 2 weeks. So they looks fine. And when we check the their ionocyte number and the gene expression of the zebrafish gill, we did find that the ion transporter responsible for the uptake of the calcium and chloride and sodium, the expression is actually decreased, and the cell number also decreased. Yeah, that is our finding.
Okay, so if there's no more question, we'll take some break. So we'll come back at 10:25 for student presentation. Okay.
Thank you.
Okay, we'll start today's student presentation.
Hello, my name is Zhang Yu, and she is Zhang Yizhen. We will propose a concentration state dependence regulation of independent cell concerned by the steno cause 1A 1I 控制。In organs, the normal functions and the survival of cell not only rely on the cell development and the proliferation, but also affected by the quiescent state of the cell. The quiescent state of the cell helps prevent the uncontrolled cell proliferation and maintain the cell functions and structure. Therefore, how to control the cell switch between the cell division and the quiescence is important. In a recent study, it has been shown that the insulin PI3 kinase (AKT) and mTOR signaling pathway play a key role in regulate quiescent cell perfusion. In mouse study, it can reactivate the neuron stem cell and the intestinal stem cell by the IGF-2. Although the IGF signal pathway play an important role in the regulation of quiescent proliferation, how the hormone pathway to advance in a cell type manner remain a question. To address this issue, the other developer liver-fish model uses the NAR cell, which can compensate of the absorbing calcium ion from the surrounding to maintain the calcium stability in the fish. When the water rich in the calcium ions, NAR cells are in quiescent state. But when the calcium ions are reduced, the cell quickly enter the advanced cell cycles and the proliferation. Small mouse, the transition of NAR cell from the quiescent to the proliferation is mediated by cell cost species advance of the GIF signaling pathway. In addition, the proliferation study a metaprocess called PAPPA was a higher expression in the NAR cells could control the binding of the NAR to the cell from the upstream. Therefore, the author want to understand the mechanism between the calcium ion concentrations and the AKT pathway. To understand this mechanism, the author focus on the STC1A. This protein are related to the regulation of the calcium ions resorbing in the fish, and in some mammal studies show the ITC1 and ITC2 can bind in the PAPP and inhibit its functions. Based on those observations, the author asked the two questions: where the constant ion level in the body are regulation of the influence of STC1, and whether one or more STC protein regulation NAR cell quiescent loss PAPP. First, in order to determine whether the calcium ions concentrations affect the exposure of the STC, rebarbished embryos were controlled with the different concentration of the calcium ions. Only the NRI level of the STC1 increased with the increased calcium ions concentration, where the other STC did not increase significant. Next, the others use the PAPP homologous mutant to study the impact the calcium ions level in the body. The mutant fish all suffer from severe calcium deficiency, and the calcium ion level in the body are significantly reduced compared with the wild type. It can be seen from the figures that almost of STC1 mRNA in the PAPP homologous mutant fish is significantly reduced, where other STC are not. This data indicate the STC1 gene expression is regulation in the calcium ions dependent manner in the liver fish inputs. To determine the in vivo function of the STC1A, 3 STC1A mutant fish live will genetically use the CRISPR-Cas9, known as STC1A plus 17, STC1A delta 18 plus 1, and the STC1A delta 35. The purchase of the STC1A heterologous cross where the mix of the homologous, heterologous, and the wild type. This one is wild type, this one is heterologous, and this one is homologous mutant. And then we'll reach an advance in the double bond manner. No difference in the body size, development rate, and over morphologies will detect almost the 3 genotype until the 3 DPF, at least. But start at the 4 DPF, many STC1A homologous mutant fish show a hard demand, which became a more significant at the 5 DPF. 5 DPF. This is not infallible swing better there either. By day 6 and 7, the mutant fish develop 7 bodies, strengthen, and reduce survival rate. This finds that although the STC1A did not affect the embryos, patterns, and ground, it can cause for the larva's survival. Next, the others measure the calcium ions level in the fish. The body's calcium ions will significantly increase in the STC1A. Homologous mutants compared with the control. Next, to the insert the possible change in the NAR cell in the NAR cell ensure the hydrophobic and fluorescence labeling of the NAR cell in the heterologous, homologous, and wild type. STC1A homologous mutants fish have the significant more NAR cells than other fish. You can see the cell is more. This one and this one. Quantum developed reduce also show the NAR cell in the STC1A double mutant fish develop more times. You can see the cell is more. STC1A homologous mutant fish have significant more NAR cells than other fish. Therefore, NAR cells in the STC1A homologous mutant fish were often observed in cell culture and address a new cell. This fund indicates the genomic delete of the STC1A result in the increased NAR cells proliferation and the elevation calcium level in the body.
Okay, then next, we're going to move on the next result. So as we know, based on previous studies, PAPPAA plays a relevant role in the STC1A regulated pathway. And as shown in the figure A, we can see that the mRNA level of PAPPAA significantly increased in the STC1A homozygous mutant genotype, and it indicates that STC1A highly correlates with PAPPAA expression. And as for figure B, the knockdown of STC1A, the knockdown of STC1A by the STC1AR gRNAs result in more NAR cells number. And as you can see in figure B, that the DMSO is the control group, and the DMSO with gRNAs injected is the group that STC1A gene has been knocked down. And the NAR cells number is way more significantly more than the DMSO control group. And as for other three groups, we can see that since BMS is a IGF receptor inhibitor, it blocks the IGF receptor pathway, and it does not allow further IGF signaling. And for zinc chloride and betimastat, these two are the PAPPAA inhibitors. So it kind of like block further mechanism as well. And as for figure 3, we can see that since the NR cell quiescence proliferation is mediated by PAPPAA, the knockdown of PAPPAA doesn't allow the NR cells to increase. And as for this result, likewise, STC1A knockdown had no effect on NR cell numbers under the absence of IGF binding protein 5A. And after confirming the roles of PAPPAA and IGF binding protein 5A in NAR quiescence, they further measured NR cells with double mutants. The double mutant is the For example, in figure E, the double mutant is STC1A and PAPBAA, and in figure F, it's about STC1A and IGF binding protein. With double mutants, the results suggest that STC1A promotes NAR cell quiescence proliferation balance dependent on PAPBAA and IGF binding protein 5A. And then so far, the regulation loop before IGF attached to IGF-1 receptor has been built up. As known, there are several components throughout the IGF signaling pathway. And after the IGF binds to the receptor, it activates IGF-1 receptor-mediated PI3 kinase AKT TOR signaling. It's a very long name, so I will just call it IGF signaling. In STC1A homozygous mutant fish, the number of phosphorylated AKT positive cells increase. As you see, the AKT phosphorylation is essential throughout the IGF signaling. Yeah. So the number of phosphorylated AKT positive cells increased compared to the others. And such an effect was abolished after inhibiting IGF receptors, since the pathway will be blocked partially or completely, and no AKT phosphorylation will be allowed under these circumstances. And besides inhibiting IGF receptor, the inhibitors functioning on other components throughout the IGF signaling were also applied. As a result, and you can see in the figure B, I think it's quite obvious, and they have kind of calculated up all of the NAR cells through in each experiment. As a result, it is quite obvious that treatments with warfarin and MK206 and rapamycin all decrease cell proliferation if compared with the DMSO treatment. And all of these, the BMS, the warfarin and MK206 and rapamycin, they are all inhibitors of each component throughout the IGF pathway. So while U0126 treatments show no difference since it inhibits the MMP kinase, and this kinase does not play a part in the IGF receptor AKT signaling. So back to the phenotypes that we mentioned before. Cardiac edema, body swelling, and premature death appeared in STC1A knockdown fishes, and which were unexpected to the authors. And they postulated that this may be a result of abnormal quiescence proliferation balance, and that STC1A homozygous mutant fish had an elevated death rate as well. So, however, STC1A and PAPBAA double mutant fish not only show none of these abnormal phenotypes, their survival rate was also similar to those of the wild types. So that, likewise, the knockdown of IGF binding protein 5A displayed a similar result. And then the BMS treatment that inhibited IGF-1 receptor and TOR signaling inhibition also eliminated fish from those abnormal phenotypes. And it proves that those phenotypes in STC1A knockdown fish occur due to elevated IGF signaling and NAR cell quiescence proliferation imbalance. In conclusion, this is the completed model they proposed. So in the left figure, the STC under normal calcium conditions, STC1A is expressed and inhibited PAPPA IGF binding protein 5A proteolysis. While under low calcium conditions, STC1A expression is reduced, thus activates PAPPA IGF binding protein proteolysis. And in this case, the IGF will be released, and then it can be bind to the IGF receptor, and thus the IGF signaling pathway will be activated. In the middle figure, you can see that there's low calcium uptake. This is because the calcium concentration in the environment is already quite low. So when the NR cell taking more calcium, it will result in the low calcium uptake. And in the as for the STC1A mutant fish, the loss of STC1A leads to IGF binding protein 5A proteolysis, and then activates the IGF signaling. It's quite similar to the wild type fish when it's in low calcium concentration, but it is just the STC1 is completely absent in this condition. So, in fact, many of the mechanisms mentioned in this research were already solved in previous studies. The blue blocks is the facts that we already know before. But throughout this studies, we now understand that that in fact the STC1 expression is associated with the PAPPA mediated IGF binding protein 5A proteolysis. And this is not only about the STC1 expression. It is only merely about one of the genes inside the STC1A expression. So what's the importance of this study? It completed the mechanism of the NR cell quiescence, and it provides us more details about this cell quiescence and proliferation balance. Then now we can move on to the further studies. In zebrafish, the STC1A mediates a series of pathways that leads to cell proliferation or quiescence and cause some abnormal phenotypes we mentioned before. For example, the deficiency of heart development, or there's a trouble about some cardiac muscle function. So what about mammals? The authors have mentioned that a similar loop is conserved in mammals, which is regulated by a chronic disease gene, STC1. And the deficiency, all of those components regulated by STC1 is listed on the slides that you can see. There are IGF-1 and IGF-1 receptor, IGF binding protein 5, and the PAPPA. It's quite familiar, right? So, and this is deficiency of the IGF signaling leads to abnormal kidney function in mammals. So in this model we show on the screen is that when there's overexpression of those components, it will lead to a disease, a kidney disease in mice. That there will be worse kidney functions, some kidney injuries, and their kidney will be puffed up. There's a lot of sites growing on the kidneys. But when the PAPPA is knocked out, it'll be decreased sites development and then improved kidney injury response. And now, throughout this model, in fact, the NR cells in zebrafish are functionally and molecularly similar to human renal epithelial cells. So, if the first studies led allow us to understand more about this pathway and all of the mechanism about the consensus proliferation balance, not only in zebrafish, but only in mammal renal cells. It may also provide some possible treatments for renal disease. Yeah. Thank you. Yeah, any questions?
I have a naive question. So what is calcium? Can you give us a brief introduction, what does the calcium coming from, secreted by which organ, and do you have any information of the calcium receptors? Thank you.
这个问就是 STC 它是什么东西？它从哪里来？就是你之前有删掉 PPT 上面有写到的东西。
STC is a protein found in the tennis corpuscle of the Tortoise fish. STC receptor. This I don't know. STC receptor. In this paper, the STC receptor is a PAPPA.
Do you want to say calcium receptors are not discovered yet?
I'm not sure. Oh, yes.
Okay, thank you.
Any questions?
I guess someone has to ask a question from Kyoto, so I'll ask. In the drug treatments with BSM and Wortmannin, it looks like in the mutant cells, mutant organisms, you normalized levels, you normalized numbers of cells, but why didn't the drug treatment reduce them below normal cell levels? Maybe did they just get lucky with their dose?
I'm not very sure about how the dosage they used throughout this experiment, but in my I would think that it is because it kinds of since the SAC1A gRNA injection, well, before any drug treatments, it might increase the NR cells, but when partial of the receptors or PAPPA inhibits, it will just decrease some of them, but not making the NR cells number lower than they originally have.
Okay.
Yeah.
And I think I just wanted to point out the difference between quiescence and actually cell death. So the cells apparently aren't dying. They're just staying in steady state. Okay, thank you.
Okay, so if there's no more question, I think we will end this week's lecture. So we'll see you next time. I think next week is a holiday, so next week we will not have a lecture. So we'll see you 2 weeks from today.
Thank you very much.
Thank you. Bye bye.
Lee?